Welcome to the deep dive. Today we're tackling a really significant health issue, peripheral arterial disease, or PAD.
Right, and specifically when it gets pretty serious, progressing to critical limb ischemia, CLI.
Exactly, CLI. That's where you see severe pain, tissue loss. It's a real threat to the limbs.
It is, but the body, it's not just sitting there. It has these amazing built-in repair systems.
Yeah, like arteriogenesis,
Yeah,
the body building its own bypasses,
precisely. Arteriogenesis creates new routes around blockages, and then there's angiogenesis.
which is more like sprouting tiny new capillaries, right, to get blood to those starved tissues.
Exactly. It's really the body's way of trying to cope. Understanding these processes is, well, it's fundamental if we want help when they aren't quite enough.
Because they aren't always enough, are they? Despite these natural mechanisms, CLI can lead to really serious outcomes. Amputation, obviously, but also a huge drop in quality of life, and it costs the healthcare system a fortune.
Absolutely. And we have the traditional ways to manage PAD, lifestyle changes, exercise, meds.
And sometimes more invasive things like endovascular procedures or surgery.
Those are the mainstays, definitely. But the problem is, for some folks with CLI, those options just aren't on the table. Maybe they're too sick or the disease is too advanced.
And that's really where we're focusing today, isn't it? This whole area of biologic treatments.
Yes, we're going to dive into things like gene-based therapies, molecular therapies, cell-based therapies, basically looking for ways to boost that natural healing process and hopefully save limbs.
It sounds like the journey here hasn't been straightforward, though.
Oh, not at all. Initially, there was a lot of buzz around gene and molecular stuff, especially growth factors, things like VEGF and FGF.
Okay, VEGF, vascular endothelial growth factor, and FGF, fibroblast growth factor.
Hmm.
We know they promote vessel growth, so why weren't they the answer?
Well, it's interesting. The early, smaller studies, they looked promising. You'd see more collateral vessels, maybe some improvement in blood flow numbers. But when they scaled up to bigger trials, the results were often, well, mixed. They didn't consistently see big improvements in things that really matter to patients, like walking further or avoiding amputation. Plus, sometimes there were side effects, like swelling.
So just pumping in more growth factors wasn't the magic bullet,
It seems not. It kind of suggested the problem might be more complicated than just a lack of these signals.
which I guess leads us to the cell-based approaches. You mentioned things like EPCs and BMMNCs.
Right. Endothelial progenitor cells, EPCs, and bone marrow-derived mononuclear cells, BMMNCs. These have definitely generated more excitement recently.
We see more promise.
The evidence is building, yeah. It looks like these approaches might offer real benefits. So that's our mission for this deep dive. Get into the science of neovascularization, that's arteriogenesis, and angiogenesis, understand how they should work, and then see what the human trials of these biologic therapies are actually telling us. We want you to come away with a clear picture of the key concepts and where the breakthroughs might be.
Sounds like a plan. So let's start with that foundation. Neovascularization, the body's repair kit. Give us the overview.
Okay. So neovascularization is just the umbrella term for making new blood vessels. It really covers three main processes, though two are most relevant for us today.
And those are
Well, there's vasculogenesis, arteriogenesis, and angiogenesis.
vasculogenesis. What's that one about?
That's primarily about the initial formation of blood vessels, like when an embryo is developing.
Yeah.
It lays down the first network from scratch using progenitor cells. It happens mostly then, but there's maybe some role for it in adults too, in adaptation.
Okay, the first draft, so to speak. But for dealing with blockages later in life, we're mainly talking arteriogenesis and angiogenesis.
Exactly. Those are the key adaptive responses we're interested in for PAD and CLI.
All right, let's tackle arteriogenesis first, building those bypass roads. How does the body actually pull off that kind of major construction project?
It's pretty amazing, actually. Arteriogenesis is all about taking existing small connections between arteries, think tiny side streets, and growing and remodeling them into much larger collateral vessels. These become the effective detours around the main blockage.
And what kicks it off? What's the signal to start building?
It's mainly about physics, really. Changes in blood flow dynamics, specifically something called fluid shear stress, or FS.
Fluid shear stress.
Yeah, when a main artery gets narrowed by plaque, the blood has to squeeze through the remaining space faster, or it gets shunted into those small connecting vessels. This increased flow speed and pressure, the stress on the walls of those small vessels, that's the primary trigger.
So the extra pressure in the side streets is the alarm bell. What happens then?
That increased FSS wakes up the endothelial cells, the cells lining the inside of those collateral vessels. They get activated and start churning out a whole bunch of proteins needed for vessel growth. It's not just one thing. It's a complex mix: HIF1, EGR1, VEGF, FGF, various inflammatory signals, you name it. Think of it as mobilizing the entire construction crew and materials list.
Wow, okay, so the crew's assembled, blood's being pushed into these small vessels. How do they actually get bigger and turn into useful bypasses?
Well, first, the endothelial cells release nitric oxide, NO.
Nitric oxide right there relaxes blood vessels.
Exactly. It causes vasodilation, widens the vessel, which immediately improves flow a bit. But NO also stimulates the release of VEGF.
Ah, VEGF again. So it's a positive feedback loop.
Sort of, yeah. VEGF then triggers the release of other molecules, like cell adhesion molecules, enzymes, and a chemical attractant called MCP1. This makes the inner lining of the vessel kind of sticky.
Sticky? Why would you want a sticky blood vessel?
It sounds weird, but it's to attract immune cells, specifically monocytes, from the blood.
Immune cells. What are they doing in vessel construction?
That's a great point. They're not just defenders here. These monocytes stick to the cams, squeeze into the vessel wall, or gather on the outside. Once there, they release more signaling molecules like TNF-alpha, which actually calls in more monocytes. It's like they're amplifying the signal.
Okay, so a swarm of monocytes arrives. Then what?
Then these monocytes and related cells called macrophages release a whole cocktail of growth factors, chemokines, cytokines, and crucially enzymes called matrix metalloproteinases, or MMPs.
MMPs. Those break down tissue, right?
They do. They help break down the existing structure a bit, allowing space for remodeling and growth. And all those growth factors stimulate the cells within the vessel wall, particularly the smooth muscle cells, to multiply and change. So the monocytes are directing the demolition and reconstruction in a way.
So the immune system is actively reshaping the vessel. What about the smooth muscle cells themselves?
They definitely respond. They proliferate, change their behavior. And there's another mechanical force, the shear pressure inside the widening vessel, called circumferential wall stress. This also tells the smooth muscle cells to grow and thicken the wall, making it stronger to handle the increased flow.
Right, reinforcing the structure. So you get all this growth.
Yeah.
Does it just keep going? Or is there some refinement?
Oh, absolutely. The final phase involves pruning. The body sort of assesses the new network it's built. Smaller, less efficient collaterals that aren't carrying much flow tend to regress and disappear.
Like closing off the less useful side roads once the main bypass is working.
Exactly. You end up with a few larger, more robust and efficient collateral arteries doing the heavy lifting. It streamlines the system.
That makes a lot of sense. Now, critically, you mentioned that cardiovascular risk factors mess this process up. That must be a big reason why this natural system fails in people with CLI.
It absolutely is. Each risk factor throws a wrench in the works in different ways. Aging itself is a factor. Older individuals tend to have lower nitric oxide, less response to low oxygen via HIF, reduced levels of key growth factors. The whole system is just less responsive.
Okay, so aging slows it down. What about diabetes?
Diabetes is a big one. It impairs the body's ability to get those crucial monocytes to the site, makes them less responsive to the signals calling them in, reduces the number of those helpful endothelial progenitor cells we mentioned earlier, and it messes with nitric oxide production too. It hits the process at multiple points.
Wow. And hypertension. High blood pressure.
That one's a bit tricky. You'd think the higher pressure might boost the initial shear stress signal, which it might. But chronic hypertension also damages the endothelium through oxidative stress and other mechanisms, making the vessels less able to respond and grow properly over the long term.
So a double-edged sword, perhaps?
Yeah.
And high cholesterol. Hyperlipidemia.
Yeah, that gums up the works too. Bad cholesterol, the oxidized LDL, can directly interfere with how VEGF works, stop endothelial cells migrating properly, reduce levels of other factors like FGF, hinder immune cell movement, and generally impair the production of all those growth factors and adhesion molecules needed to build the vessels.
It really hits it from all angles. And smoking. We can't forget smoking.
Definitely not. Smoking is terrible for this. It hammers those endothelial progenitor cells, reduces their number, messes up their function, stops them migrating and sticking where they need to be. It also disrupts monocyte movement and decreases key signals like VEGF and HIF1.
So managing these risk factors isn't just about stopping plaque buildup.
Exactly. It's also about giving the body's own repair system, this arteriogenesis process, the best possible chance to work. The key point here is that this whole complex cascade, meeting all these signals and cell types working together, gets disrupted by these very common risk factors. That really explains why just throwing one growth factor into the mix might not be enough.
That's a fantastic explanation of arteriogenesis. Okay, let's switch gears to angiogenesis, sprouting those new capillaries. How is that different?
Angiogenesis is more focused on the micro level. It's about forming new capillaries, those tiny vessels, directly from existing ones. The main driver here isn't so much sheer stress, but lack of oxygen ischemia in the tissues themselves.
So when the tissues downstream are starved of oxygen, that's the trigger.
Pretty much. The key signal that goes up in low oxygen is hypoxia-inducible factor 1, HIF1.
HIF1 again.
Yep. And HIF1 in this context ramps up the production of VEGF.
VEGF seems to be a player in both processes then.
It is, but its main role in angiogenesis is really to make the endothelial cells in existing capillaries multiply and start sprouting outwards. It also makes the vessels a bit more leaky, which might help cells and nutrients get out.
And you mentioned MMPs earlier, the enzymes that break down tissue.
They're important here too. They get activated and chew away at the basement membrane and the surrounding matrix, basically clearing a path for these new capillary sprouts to grow into the tissue and hopefully improve oxygen delivery.
So VEGF signals grow, MMPs clear the path, but you said this often isn't enough for major blockages.
Right. Angiogenesis increases the density of capillaries, which is good for local exchange, getting oxygen a tiny bit further. But it doesn't typically build a big enough pipe to bypass a major blockage upstream like arteriogenesis can. They're both important, often happening together, but they serve slightly different functions in response to the blockage.
Got it. So how does this sprouting actually work at the cellular level? What are the mechanics?
Well, the main mechanism is called sprouting angiogenesis. Imagine an existing capillary. Some endothelial cells get the signal and start pushing outwards into the tissue. The basement membrane around them breaks down.
And then you mentioned specialized cells. Tip, stalk, phalanx.
Exactly. It's like a little construction team within the sprout. At the very front is the tip cell. It's migratory, extending these little feelers called filopodia, sensing the VEGF signals, kind of guiding the way. It doesn't divide much itself.
Okay, the leader. Who's behind it?
Behind the tip cell are the stalk cells. These guys are the workers. They do divide rapidly, making the sprout longer. They also start forming the actual hollow tube, the lumen, and they lay down the new basement membrane around the sprout.
So they build the structure and the phalanx cells.
Once a segment of the vessel is formed, the stalk cells mature into phalanx cells. They become less active, more stable, form tight connections with their neighbors to make the vessel wall less leaky and really solidify the structure. And you also have parasites supporting cells that wrap around the capillary, helping to stabilize everything.
It sounds incredibly organized. How does a cell know whether to be a tip or a stalk?
It's regulated by a really neat signaling system involving VEGF and another pathway called Notch. Basically, VEGF encourages cells to become tip cells, but the tip cell itself produces a signal, DLL4, that activates Notch in the cells right behind it. And Notch signaling tells those cells, nope, you be stalk cells. It's called lateral inhibition. It ensures you only have a few leaders and lots of followers building the vessel.
That's clever. A self-organizing system. And forming the hollow tube, the lumen. How's that done?
That's tubulogenesis. It's about establishing that inside-outside polarity. There are a few ideas on how it happens. Maybe internal vacuoles, these little fluid sacs, fuse together inside the cells, or maybe vacuoles are released between cells and merge. Another idea involves rearranging the connections between cells. VEGF and blood flow itself probably help expand that lumen once it starts forming.
Okay, multiple ways to get that tube built. Now, you also mentioned intussusceptive angiogenesis. Non-sprouting. What on earth is that?
Yeah, it's quite different. Intussusception means growth from within. Instead of sprouting outwards, an existing capillary essentially splits down the middle to form two.
It splits itself. How?
Imagine the opposite walls of a capillary touching inside. Then a sort of pillar of tissue containing matrix and supporting cells grows across that contact point, eventually dividing the original vessel into two parallel ones.
Wow, so it doubles the vessels in place. That sounds really efficient.
It is. It's a very quick way to increase the density of the capillary network and the surface area for exchanging oxygen and nutrients without needing lots of new cell division or disrupting the surrounding tissue much.
Are there different kinds of this intussusception?
Yeah, they describe a few variations based on how it reorganizes the network. Some mainly increase surface area, others help form slightly larger microvessels like arterioles and venules out of capillaries, and some optimize the angles where vessels branch off to improve blood flow efficiency based on shear stress.
Fascinating. So it's more about optimizing the existing network structure.
Exactly. And its benefits are minimal cell division, less leakiness compared to sprouting, and less tissue disturbance. Now, once you have all these new vessels, whether from sprouting or intussusception, the network doesn't just stay static.
Right, you mentioned remodeling and pruning.
Yes, the whole capillary bed is constantly adapting. Vessels in areas with high metabolic demand and good blood flow tend to grow and stabilize, but vessels in areas with low flow, low shear stress, maybe less VEGF signaling, they can actually regress and disappear. That's the pruning part. It's all about optimizing the network to match the tissue's needs. And the vessels mature, getting stronger and less leaky through interactions between all the different cell types and the matrix around them.
It's just incredibly dynamic and complex, which I suppose brings us back to why just giving growth factors didn't work so well in CLI. It sounds like the problem might be deeper.
That's exactly the thinking now. If simply adding more VEGF or FGF didn't consistently fix the problem, maybe the issue isn't just a lack of the signal. In fact, like we said, some studies show CLI patients actually have high levels of some of these pro-angiogenic factors.
So the signals are there, maybe even shouting, but the cells aren't listening properly?
That seems to be a strong possibility. It points towards problems within the cells in the signaling pathways that are supposed to respond to those growth factors. The internal communication system might be broken.
Okay, that makes more sense. So if the growth factors themselves aren't the whole story, what else could be controlling this response? You mentioned non-coding RNAs earlier.
Yes, non-coding RNAs or ncRNAs. This is a relatively newer area, but it's adding a massive layer of complexity and potential understanding.
Right.
We're talking mainly about microRNAs, miRNAs, and long non-coding RNAs, LNCRNAs.
Non-coding RNAs.
Mhm.
But they don't make proteins, right? What do they do?
Exactly. MicroRNAs are tiny little RNA strands, maybe 22 units long. They work by binding to messenger RNAs, the templates for proteins, and usually blocking them from being translated or causing them to be broken down. So they act like fine-tuners, controlling how much protein gets made from a gene.
So they can dial protein production up or down.
Effectively, yes, mostly down by interfering with the mRNA. Long, non-coding RNAs are much bigger, over 200 units, and they do all sorts of things, influence how DNA is packaged and read, affect mRNA stability, interact with proteins. They're involved in lots of regulatory processes.
And these ncRNAs are involved in blood vessel growth.
Oh, absolutely. They're emerging as key regulators of all those signaling pathways we've been talking about. And what's really complex is that a single microRNA can influence multiple targets. It might promote one aspect of vessel growth while inhibiting another, depending on the context.
Can you give an example?
Sure. There's one called miR-126. It's very common in endothelial cells. It can actually inhibit angiogenesis slightly by reducing a molecule called VCM1, which helps immune cells stick. But it can also promote angiogenesis by blocking other molecules that normally put the brakes on VEGF signaling. So it's doing two seemingly opposite things.
Wow, okay. That is complex.
Yeah. Or miR-155, which seems to have some anti-angiogenic effects but might be pro-arteriogenic. There are whole families of these things involved. It really highlights why the simple, more growth factor, more vessels idea was probably too simple for a condition like CLI, where these regulatory networks might be messed up.
And the long non-coding RNAs.
Their roles are even more diverse and still being figured out. But yes, specific LNCRNAs have been shown in experiments to either promote or inhibit neovascularization by influencing various genes and pathways.
So it's not just the signals, but how the cells interpret those signals. And these ncRNAs are like the interpreters or maybe the volume controls.
Yeah, that's a good way to think about it. And the idea is that in CLI, maybe these ncRNA regulators aren't working right, contributing to the poor vessel growth even when growth factors are around. It's a really active area of research.
This really changes the picture. Okay, so with all this complexity in mind, let's turn to the clinical trials. How has this science actually been translated into attempted treatments for the CLI patients who can't get surgery?
Right. This is where the rubber meets the road. For these patients, boosting neovascularization with biologic therapies, genes, proteins, cells has been the big hope. So, bearing in mind all the complexities we just discussed, let's look at how these approaches have actually performed in humans.
And we started by saying the gene and protein therapies, mostly the growth factors, had kind of mixed results. Can you walk us through some of those? VEGF first.
Okay, VEGF. Logically, it was the first big target, early small phase I trials. They did show some encouraging signs. Imaging showed maybe more collateral vessels, blood flow measures like the ABI sometimes improved, some wounds healed better, maybe even a limb saved here and there. But there was also that side effect of peripheral edema, swelling.
Okay, some promise, some problems. What about bigger trials?
The larger phase 2 trials, testing different delivery methods, injecting into arteries or muscle, were really quite mixed. Some showed improvements in ABI or wound healing, maybe more so in diabetic patients in some studies. But overall, they didn't consistently hit the big endpoints like significantly better exercise tolerance or reliably lower amputation rates across the board. The focus with VEGF was primarily boosting angiogenesis, those small vessels.
So initial hope didn't fully translate. What about FGF, fibroblast growth factor, another big player, right? Stimulates both angiogenesis and arteriogenesis.
Yeah, FGF was tried too in several phase 2 and 3 trials. Again, some studies reported patients could walk further or had better ABI scores. One phase 2 trial, Talisman, even hinted at fewer amputations, which was exciting, although it didn't show much effect on ulcer healing.
Fewer amputations sounds good.
It did, but unfortunately, the big follow-up phase 3 trial, Tamaris, couldn't replicate that finding. It just didn't show a significant reduction in amputations.
Ah, it's tough. A familiar story in research, I guess. Any other growth factors tried? HGF?
Hepatocyte growth factor, HGF, yes. It helps endothelial cells function and multiply. Again, some phase 2 trials looked okay. Maybe better oxygen levels in the skin, TCPO2, some improved ulcer healing, better quality of life reported in some cases. But like the others, no consistent big wins on ABI or amputation rates. There was a newer plasmid version, VM202, that showed some early promise.
It really seems like this single-factor approach was challenging. Any other gene or protein ideas that were tested?
A couple more worth mentioning. DEL1, that's developmentally regulated endothelial locus 1, it's a protein involved in the vessel matrix. A phase III trial actually looked pretty positive, reporting improvements in exercise tolerance, time to leg pain, ABI, and quality of life.
Okay, that sounds a bit better.
It did, but it's still relatively early data. On the other hand, they tried gene therapy to boost HIF1, the hypoxia factor, hoping that would trigger the natural response. But a phase 2 trial in patients with claudication, which is less severe than CLI, showed no benefit in walking or other outcomes.
So overall, the track record for gene and protein therapies based on single factors is, well, not stellar for CLI.
It's been challenging, yes, which is why the focus increasingly shifted towards cell-based therapies. As we said, these seem to be generating more encouraging results overall.
Right, and the main players there are endothelial progenitor cells, EPCs, and bone marrow-derived cells, BMCs.
Those are the main ones, yes. EPCs are precursors that can become endothelial cells, the vessel lining. BMCs are a richer mix from the bone marrow, including various stem and progenitor cells. Looking at the studies broadly, these cell therapies often do report improvements, better ABI, less rest pain, more ulcers healing. But the really crucial outcome, consistently saving more limbs from amputation, that's been harder to demonstrate definitively across all studies.
Okay, let's break it down. EPCs first. How have they been used?
A couple of ways. One idea was to use a drug, rhGM-CSF, to coax the patient's own EPCs out of their bone marrow into the blood. A trial called START did this. It showed increased blood flow in the tiny vessels, the microcirculation, but didn't actually improve ABI or exercise ability.
So just mobilizing them wasn't enough. What about giving the cells directly?
That seems potentially better. One trial injected CD34 plus EPCs directly into the leg muscles. They saw improvements in pain, walking distance, wound healing, and blood flow measures. Another group used CD133 plus EPCs, a different marker, and reported preventing amputation in some patients and improving quality of life. Injecting CD133 plus cells into the artery also showed benefits in ABI, oxygen levels, and disease severity, especially if there was already some flow in the lower leg arteries.
Okay, so direct delivery of EPCs looks more promising, maybe especially certain types or in certain situations. What about the bone marrow-derived cells, the BMCs, the broader mix?
BMCs, often used as the mononuclear cell fraction, BMMNCs, have probably been studied the most. Many phase 2 trials injected these either into the artery or muscle, and they frequently reported good signs: ulcers healing better, less rest pain, longer time before needing another procedure, better amputation-free survival in some cohorts, improved walking, better ABI. Lots of positive signals.
That sounds generally more positive than the single growth factors.
Yeah. It does seem that way. One meta-analysis, pooling data from multiple studies, even suggested injecting into the muscle might be better than using cells mobilized from peripheral blood with GCSF, and that BMCs might be better overall. And significantly, there was a phase 3 trial with BMMNCs that reported improvements across the board: pain, quality of life, ABI, disease stage, and fewer amputations.
A phase 3 trial showing fewer amputations. That sounds important.
It is potentially very important. However, it's worth noting that a big Cochrane review, looking systematically at all the evidence, concluded that while there were positive trends, there weren't yet clear, definitive differences between using bone marrow cells versus mobilized peripheral blood cells, or between injecting into artery versus muscle for the major outcomes like amputation. They cautioned many studies were small, maybe not powerful enough to pick up real differences.
So promising signals, especially with BMCs, but maybe not quite slam dunk evidence across the board yet. Still needs more work.
That's a fair summary. The consensus is building, but more robust data is needed to really nail down the best cell type, the best delivery route, and which patients benefit most.
Got it. Were there any other cell types tried? You mentioned fully differentiated cells.
Yes, a slightly different tack. One early trial used a mix of smooth muscle cells engineered to produce VEGF and endothelial cells engineered to produce Ang1, another vessel-stabilizing factor. This multi-gene angio approach showed increased walking time. A follow-up phase A trial reported a pretty good 72% amputation-free survival at 1 year.
That's interesting, too, using cells as little drug factories right where they're needed. So the cell therapy field is definitely active. Any big trials ongoing we should watch out for?
Yeah, definitely. There are at least 2 phase 3 trials actively recruiting or ongoing that are key. One is the MAGI-SKELTA trial, directly comparing bone marrow MNCs against peripheral blood MNCs, both injected into muscle. The other is the DONG trial, looking at purified CD34 plus EPCs versus peripheral blood MNCs. Results from these larger trials will be really important for guiding clinical practice.
Okay, this has been incredibly comprehensive. We've gone from the basic biology of how blood vessels form and adapt, through the challenges with early therapies, to the current promise and remaining questions around cell-based approaches. Let's try to summarize the big takeaways.
Absolutely. So number 1, the body has these amazing natural processes, arteriogenesis and angiogenesis, to try and compensate for blocked arteries in PA and CLI. 2, early biologic therapies trying to just boost single growth factors like VEGF or FGF generally had limited success in major clinical outcomes, likely because the underlying problem in CLI is more complex than just a lack of those signals.
Right, the signaling pathways themselves might be impaired, and those non-coding RNAs are emerging as potentially key regulators, adding another layer of complexity.
Precisely. 3, cell-based therapies using endothelial progenitors, or perhaps more effectively mixed bone marrow cells, are showing more consistent promise. We're seeing improvements in blood flow, pain, ulcer healing, and potentially even limb salvage in some studies, though more definitive proof from large trials is still needed.
And it's clear this is still very much an evolving field. Cell therapies seem closest to potentially becoming a standard option for these no-option patients, but optimizing them is key, and understanding those ncRNAs could open up totally new therapeutic avenues down the line.
Exactly. Which leads us to maybe a final thought for you, the listener, to consider. Given how incredibly complex neovascularization is and how different CLI can be from person to person, what might the real future of treatment look like? Could we move towards truly personalized biologic therapies?
Personalized, meaning?
Meaning, could we one day analyze an individual patient's specific molecular profile, their growth factor levels, their ncRNA patterns, their cell function, and then design a therapy tailored specifically for them? Maybe it's a specific cell type, maybe it's targeting a particular microRNA, maybe it's a combination.
Yeah.
A treatment designed not just to flood the system, but to precisely tune their own body's healing potential. Perhaps that's the ultimate goal, unlocking and fine-tuning the individual's intrinsic repair capacity.